Proteins are polymers of amino acids, and we consider the primary structure of proteins to be the amino acid sequence. When two amino acids combine together, the amino acids being the monomers, they would form a dimer. If three come together, it'd be a trimer, and so on and so forth, up until you get proteins. When two or more amino acids combine together, they form what's called peptide bonds. And peptide bonds are essentially just the connector between two individual amino acids. And as you'll see in the picture, they ultimately form an amide. So we have here on the left glycine, which has that carboxylate group. And on the right, we have alanine, which has that ammonium group. We learned in the amines and amides chapter that when you have a carboxylic acid and it reacts with an amine, they form an amide, as you can see, as I'm going to show here in this drawing. So this is just a refresher from the previous chapter, but a carboxylic acid plus some amine. I'll just use ammonia here, even though it's technically not an amine. It'll serve its purpose, where we get the carbonyl, not oxygen, we get the nitrogen, it'll be NH2 here, and we cleave off water. So you can see in the above reaction, it's the same thing. Technically this has the carboxylate and an ammonium, but overall we're going to get the same result from this. Water is cleaved off. We have the formation of our amide right there. So peptide bonds can be defined as the bond that links the two amino acids together. So as I've written there, the carboxylate group of one amino acid reacts with the ammonium group of another amino acid to form an amide. These amide bonds are classified as peptide bonds. Peptides, when we're not talking about bonds, peptides are sequences of amino acids that are small, between 2 to 50 amino acids in length. Anything more than 50 amino acids would be considered a protein. Anything less than 2 would just be a single amino acid. So in this slide we have two amino acids, glycine and alanine. They will combine together to form a dimer of amino acids, as it shows over here, glycylalanine. What happens is the carboxylate group of glycine will react with the ammonium portion of the alanine. And again, it will kick off water as a product of that reaction. So you can see here on the right, you have the carboxylate portion from the glycine and the ammonium portion from the alanine. The bond right here in the middle is considered the peptide bond. But again, this whole group is considered an amide in organic chemistry. Okay, and so what we get there is a dimer, again, between glycine and alanine. That glycine would be on the left, alanine is on the right. When any number of amino acids link together like this to form one or more peptide bonds, we're always left with a free ammonium on the left and a free carboxylate on the right there. Okay, and that's always going to be true. Now this is just a dimer, so we have one of each, and then one amide. But if we had a trimer, well, then we'd have some NH3, then we'd have an R here, the carbonyl, and then you've got your peptide bond. Then in this case, you have that this would be an R group. If we had a third amino acid, it would be right here. Kind of running out of space, but then you'd have O minus there. So we would have one amino acid right here. We'd have a second amino acid right there in red, and then a third one in blue. But in each case, no matter if it's a dimer, a trimer, or anything up to, you know, 1,000,000 or what have you, we would have a free ammonium, and I should put the charge there. That's an NH3 plus right there. So free ammonium, and then we have a free carboxylate on one end. Okay, and that's going to happen no matter what. We're always going to have those. So now between the glycine and the alanine, we could have the glycine react its carboxylate with the ammonium of alanine, or it could be reversed. So technically you have this species here, the glycine. It could react instead in this orientation right here. And that would give us a different peptide or a different dimer of the alanine and glycine. So in the picture that was given to us, we had glycyl alanine as the dimer. In this one that I've changed, it would be alanyl glycine. Alanine would be first, and then the glycine would be second. And so, you know, with every subsequent additional amino acid, you're going to get many more possible different sequences of the peptide chain or the protein. For example, if there are 2 amino acids, you have the 2 possibilities. If there's 3 amino acids, you'll get 6 different possibilities. If there's 4 amino acids, there's 24 different possibilities. And if there's 5 amino acids, it jumps up to 60. So it's growing at an increasing rate, so it's exponential. And so you can imagine that since the proteins are kind of the doers of our bodies, or things that need to be built in specific ways, getting them to align properly to form the correct amino acids is extremely challenging. When we're looking at proteins or peptides, we want to be able to figure out the beginning and the end of the sequence. And it turns out that for arbitrary reasons, the N-Terminus is the beginning and the C-Terminus is the end. The N-Terminus is identifiable by looking for the free ammonium ion, which every end of every amino acid sequence will have that. The C-Terminus can be identified by looking for the free carboxylate ion. And again, every single sequence of amino acids will have that. So as you can see, the N-terminus here, as I've defined it, is the beginning of an amino acid sequence. It is the terminus of the amino acid sequence that has the free ammonium ion. The C-terminus of an amino acid sequence is the terminus that has the free carboxylate ion, the CO2 minus. So we have the N-terminus in blue there, and the C-terminus in red. The peptide bond is the green bond I have there. That's also, again, the amide bond. So you'd have an amide. Now, if we were to be connecting a third amino acid into this chain, I could react it at the N-terminus or the C-terminus. In reality, you know, like if it was just floating around in space, it wouldn't have any reason to pick one or the other. But we could have, let me use orange here for this, or slightly orange. That carboxylate group would get changed to an amide. Hopefully you can see that. So here's the beginning of our third group. We'd have an R double prime down there, and then another carboxylate ion right there. And so that red would no longer be the C-terminus. The C-terminus would now be that the carboxylate group of that third amino acid. So if you're given a picture as like the one here at the bottom with all these ball and stick models, we can identify the amino acid sequence by looking for amide or peptide bonds. And again, the peptide is going to be between the carbonyl and the nitrogen. So you'd have one right here. We have one right here, right there, right here, right there, right there, and right there. So it's kind of helpful with all the blue and red. If we add up the number of peptide bonds, we've got 1, 2, 3, 4, 5, 6, and 7, which means we're going to have 8 amino acids linked together. And I'm going to try to highlight these for you. We have in different colors. We have this one right here going on to its nitrogen. All right. Then we'd have the second one starting at the carbonyl with its R group. That would be glycine down to its nitrogen. Then we again start at the carbonyl, go down here through to its nitrogen. Then we've got the carbonyl, the R group, the nitrogen. All of this would be part of that amino acid. And then all of this would be the next. Right there. And so on and so forth. All of this would be an amino acid, and all of this would be the last one. So if we count up the number of colors, we've got 1, 2, 3, 4, 5, 6, 7, and 8. So there are 8 amino acids. Like if you ignore this stuff at the very bottom, which tells you what they are, we should be able to identify them that way. Over here on the right and on the left ends of the chain, we have the N-Terminus due to that nitrogen being there, and the C-Terminus over here due to the carboxylate group. So the beginning of this amino acid sequence would be this amino acid, which is glycine. You can see its R group is a hydrogen. And then in the C-terminus, it's an alanine for the C-terminus. Then you could go look up and compare the structures to one of the previous slides that gave you the structures of all of the individual amino acids. But it is followed by phenylalanine, glycine, alanine, leucine, serine, and another glycine. Example 3 here asks for you to find what the amino acid at the N-terminus and the amino acid at the C-terminus is of this tripeptide. Well, for any peptide, we're always going to start with the N-terminus. So the N-terminus is PRO, which is proline, and the C-terminus will just be the complete opposite end of the chain, and so it's methionine. Next, we'll discuss how to name peptides. Peptides are drawn and named from N-terminus to C-terminus, as I mentioned. Non-terminal amino acids remove the suffix of the amino acid and replace it with YL. The amino acid at the C-terminus keeps its name. The entire name of the peptide is one word. So, for example, in this tripeptide, we have right here alanine, we have glycine, and we've got serine. Those would be the 3 amino acids. We're going to start with the N-terminus, and so it goes from alanine to alanyl. The glycine becomes glycyl, and then the serine just stays as serine. So the name of the tripeptide is alanyl glycyl serine. Now you can see in the picture they have the 3-letter abbreviations ALA, GLY, SER, separated by dashes. That's a lot easier than trying to write the whole name out, alanyl glycyl serine. And even though it is better, it's not ideal. So we have the one-letter abbreviations AGS, which is a lot quicker. So as I mentioned earlier, peptides are between 2 and 50 amino acids long, and proteins you're going to have greater than 50 amino acids. So that's just the difference, is the length of the chain. The primary structure of a protein is simply the sequence of amino acids from N-Terminus to C-Terminus. So you can see in the example at the bottom of the page, you have 3 amino acids. You've got glutamate, histidine, and proline. And so that would just be the primary structure of this amino acid, or this amino acid sequence, or this peptide. Again, you want to look for the N-terminus. So we got the NH3 plus right there, and then the C-terminus is this portion right there. So again, we do read from left to right in this case. So glutamate, histidine, and proline. Proteins can be more complicated. You can have multiple different chains attached together, as this picture is showing. In green, we have an N-terminus up here and a C-terminus down there. And then in blue, we have an N-terminus at the top and a C-terminus at the bottom. We call the individual chains the A chain and the B chain respectively. And so what you would do is you could list the A chain from N-terminus to C-terminus. So it would be glycine, isoleucine, valine, glutamine, glutamate, glutamine, cysteine, threonine, serine, isoleucine, cysteine, serine, leucine, tyrosine, etc., etc., etc. You'd go down all the way to the asparagine there at the bottom. And then for the B chain, you would start with phenylalanine, valine, asparagine, and then you go all the way down to threonine at the very bottom of that chain. And so you would just list each of them in order. Don't go across like left to right as in that, as I did there. That's not the right order for reading proteins. The following amino acids are essential amino acids, and these are ones that we cannot get from our diet. So we have histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. So again, these are amino acids we have to get from eating food. Our body cannot synthesize these in any way whatsoever. The table here shows a list of food sources and what amino acid they are deficient in. So you'll see that eggs, milk, meat, fish, and poultry, meat meaning, I guess, beef and pork, and poultry meaning anything that's a bird, they don't lack any amino acids. So it's a healthy diet just to eat meat as far as your proteins are concerned. If you eat wheat, rice, or oats, you're going to lack lysine. So let's say we have a piece or a meal with rice in it. We want to have a complete diet. We're missing lysine. You could pair that with eggs or milk, meat, fish, or poultry, obviously. And a typical thing that at least the Asians do is they eat fish a lot with rice. And that not only gives them a complete diet of amino acids, but it also gives them a complete diet of the essential lipids or fatty acids, like the omega-3 fatty acids that we talked about earlier. But let's say you don't want to eat meat or milk, you're like a vegan, I guess. Well then, if you're eating rice, okay, you got rice, you need lysine. Well, corn isn't going to help you there because it also lacks lysine. But you could have beans. Beans and rice would be a complete diet. The beans lack tryptophan and methionine, but the rice does not. So you would be getting enough. Now, if you ate beans and peas only, that would be not good. You need the methionine. If you ate just things with soy, which I don't know, that seems weird, you'd be lacking methionine. So you'd want to eat something else like wheat or oats or whatever to make sure you're getting that.